A new ultra-high-speed camera capable of capturing up to 1 trillion frames per second has revolutionized imaging, allowing scientists to visualize phenomena previously considered invisible. Unlike conventional cameras that struggle with transparent objects or ultra-fast processes, this breakthrough combines high-speed imaging with phase contrast microscopy, a technique that detects subtle variations in how light moves through different materials. By applying this to ultra-fast photography, researchers can now observe events like shockwaves in water, laser pulses traveling through crystals, and even microscopic biological changes such as neural activity in real time. The key innovation behind this system is its ability to capture single-shot images of dynamic, transparent phenomena. Traditional high-speed cameras require repeated events to compile a sequence, but this new approach, called phase-centric compressed ultrafast photography, pCUP, records everything in a single moment. This capability is crucial for studying fleeting, unrepeatable processes, such as the precise moment a nerve signal propagates or a shockwave ripples through a medium. One of the most fascinating applications of this technology is in neuroscience, where it may help visualize how nerve impulses travel through brain tissue without the need for invasive techniques or contrast agents. By detecting minute optical changes in neurons as they transmit signals, this method could provide a new way to study the brain in real time. Similarly, in combustion research, scientists can observe flame propagation and shockwave interactions at an unprecedented scale, potentially leading to advancements in energy efficiency and engine design. This camera also holds promise for studying the fundamental nature of light itself. By imaging the optical Kerr effect, a phenomenon where intense light alters the refractive index of a material, scientists can explore quantum interactions at the microscopic level. Furthermore, in fluid dynamics, this technology allows for the visualization of high-speed turbulence, cavitation bubbles, and other complex behaviors in liquids and gases. The impact of this imaging breakthrough extends across physics, engineering, medicine, and beyond. From capturing the birth of a plasma spark to analyzing the stress distribution in transparent materials, the ability to see the invisible opens new doors in scientific discovery. What other hidden processes might be revealed with this unprecedented ability to freeze time at the scale at picoseconds? This new tool could reshape our understanding of everything from fundamental physics to biological systems, making the invisible world more accessible than ever before. A newly developed ultrafast camera can capture up to 1 trillion frames per second, making it possible to record events that were previously beyond the reach of conventional imaging. Unlike standard high-speed cameras that rely on capturing multiple images sequentially, this system uses a unique method called phase-sensitive compressed ultrafast photography, pCUP. This technique enables the recording of rapid phenomena in a single shot, eliminating the need for repetitive events. The camera integrates a well-established optical principle, phase contrast microscopy, with ultrafast imaging to visualize transparent materials and light-based interactions that were previously invisible. Scientists have used this camera to capture the propagation of shock waves in water, revealing the complex dynamics of energy transfer at an ultra-small scale. Imagine a high-speed microscope that can freeze moments in time, like capturing the ripples on a pond's surface as a single drop of water falls, but at a scale and speed where even light itself slows down to reveal its behavior. Traditional cameras rely on capturing differences in light intensity to form images. However, many transparent materials and biological structures, such as thin tissues and optical waves, do not absorb enough light to create a visible contrast. This new ultrafast camera leverages phase contrast microscopy, which detects how light waves change speed and phase when passing through different materials. By analyzing these minute changes, the system translates otherwise invisible phenomena into high-contrast images. This method is particularly useful for capturing transient effects in transparent media, such as neuronal activity or shockwave propagation. Using phase contrast imaging, researchers have visualized the movement of neural signals as they travel along nerve fibers, providing insights into real-time brain activity without the need for chemical markers. Think of it like tuning a radio to pick up hidden signals in the air, except in this case, the signals are minute changes in light that reveal unseen details about the objects they pass through. Light travels at nearly 300,000 kilometers per second, making it incredibly difficult to capture its motion using conventional cameras. Most high-speed imaging systems require multiple exposures to reconstruct rapid events, but this new technology can record these phenomenon in real time with a single exposure. This capability is essential for studying ultra-fast interactions, such as laser pulses passing through different materials, explosions, or cellular processes occurring at nanosecond timescales. By capturing trillions of frames per second, researchers can observe fundamental physics in action, revealing new details about how light interacts with matter. One experiment demonstrated how a laser pulse moves through a crystalline material, showing how energy disperses at speeds close to that of light itself. It's like using a super slow magnetic camera to capture a bullet's trajectory, but instead of a bullet, the camera records pulses of light moving through space. Most high-speed cameras rely on taking multiple sequential shots to reconstruct an event. But this method fails when dealing with non-repetitive, one-time phenomena. The phase-centric ultrafast camera solves this problem using a technique called lossless encoding compressed ultrafast technology, LLEC-UP, which allows it to capture an entire ultrafast sequence in just one exposure. This single-shot capability is crucial for studying unpredictable and high-speed processes such as explosions, chemical reactions, and nerve impulses, which cannot be repeated under identical conditions. Researchers used this system to observe the rapid collapse of a cavitation bubble in water, a phenomenon that occurs too quickly to be recorded by traditional cameras. Imagine trying to take a perfect picture of a lightning strike. Standard cameras need multiple tries, but this ultra-fast camera captures the entire event in one shot, freezing every detail in time. One of the most promising applications of this technology is in the study of biological and physical shockwaves. Shockwaves. Rapid pressure changes in a medium occur in many scientific and engineering contexts, from explosions to ultrasound applications. In biology, even the electrical signals traveling through neurons cause minuscule but detectable mechanics. By capturing these ultrafast changes, researchers can investigate the behavior of shock waves in liquids, as well as potential neural activity in living tissues, without invasive procedures. The camera has been used to capture shock waves created by laser pulses in water, providing a clearer understanding of how energy propagates in fluids. Think of it like an ultra-sensitive microphone that can record sound waves far beyond human hearing, except, instead of sound, this camera detects shock waves and invisible light interactions. This new ultra-fast imaging system has wide-ranging applications in physics, engineering, and medical research. It can be used to study combustion dynamics in engines, monitor high-energy laser interactions in fusion research, or visualize neural activity in real time. Additionally, it has potential applications in material science where understanding how shock waves move through different substances can lead to stronger and more resilient materials. By integrating ultra-fast imaging with other optical techniques, researchers could develop new diagnostic tools for medical imaging and neuroscience. In future studies, scientists hope to use this technology to capture how nerve impulses travel through the brain, potentially leading to non-invasive brain imaging techniques. This is like upgrading from a grainy security camera to a high-definition slow map system. Suddenly, details that were once a blur become crystal clear, enabling new discoveries. Despite its impressive capabilities, ultrafast imaging faces several challenges. The sheer amount of data produced at 1 trillion frames per second requires advanced computational processing to reconstruct meaningful images. Additionally, achieving such high speeds requires highly sensitive sensors and advanced optics, making these cameras expensive and difficult to manufacture. Another challenge is ensuring that the imaging system does not interfere with the phenomena it observes, as ultra-fast measurements often require intense laser pulses that can alter the behavior of the system being studied. Current imaging setups are still limited in the duration they can capture due to data storage and processing constraints, but future improvements in computational algorithms may help manage these challenges. It's like building a telescope powerful enough to see exoplanets and other galaxies. The technology exists, but refining it for practical use takes time and innovation. The development of ultra-fast imaging is far from over. Future advancements will likely focus on increasing the field of view, improving resolution, and expanding applications in medicine, material science, and energy research. As computational methods evolve, additionally, integrating these cameras with AI-based analysis tools could allow real-time interpretation of ultra-fast events, unlocking new possibilities for automated scientific discovery. New designs could allow researchers to capture entire biological processes at speeds previously thought impossible, potentially leading to breakthroughs in neuroscience and drug development. This is similar to how digital photography transformed the way we capture and process images. What once required complex film development is now instant and infinitely more detailed. The development of ultra-fast imaging, capable of capturing trillions of frames per second, marks a major milestone in physics, engineering, and biology. By merging phase contrast imaging with high-speed photography, researchers can now visualize the invisible, unlocking new insights into the behavior of light, shockwaves, and biological processes. As the technology advances, its applications will continue to expand, reshaping our understanding of the physical world. This imaging breakthrough could help scientists track real-time brain activity, revolutionizing neuroscience and paving the way for non-invasive brain-computer interfaces. Just as microscopes opened up the world of cells and atoms, ultrafast imaging is revealing the previously hidden world of rapid physical and biological processes, ushering in a new age of discovery.